Good. Well, thank you all very much for coming. It's a pleasure to be your host for today. This is Kozalakidis. I'm a microbiologist by training, so this is very close to my heart. I'm the head of the laboratory services and biobanking at the International Agency for Research on Cancer. This is the in-house research agency of the World Health Organization. And we're going to talk about something that is very important. It's an important health subject that we have to do something about. And I will leave it at that because a few people and the people on this panel are doing something about it. So I would ask them to introduce themselves as they start speaking, and we'll pass over to Professor Santag to start us off. Professor, the floor is yours.
Yes, good afternoon, good evening, where everyone is. I think maybe my colleagues and you should mute so there's no echo. Try to get rid of the echo here. I'm Jerome Shentag. I'm a professor of pharmaceutical sciences. I have done antibiotic work for about 40 years now. Typically around ICU and clinical infections with pharmacokinetics and pharmacodynamics. So I feel very comfortable talking about the reality of this war, if you will. The fact is that we're losing this war. Antibiotic resistance is progressing faster than we are able to develop new antibiotics, and lately it's accelerated so that we're losing antibiotics faster than we can even begin to get them in the pipeline. And I've had a chance to reflect on this for many years, and I would like to provide what I think to be another way through this morass. And so if you could think about this as a growing threat, of course, which everybody knows. Patients die from antibiotic-resistant infections on a regular basis. By 2050, the World Health Organization has predicted that antibiotic-resistant infections will kill over 10 million people every year. That is a lot of patients because that's 22% more deaths than currently caused by cancer. So this could develop into a global pandemic very easily, and in fact, by some people has been described that way. The problem got worse during COVID. During COVID, because patients were in ICUs for a long time, many of the ICU infection pathogens developed even resistance more rapidly. So we're losing some of the newer antibiotics already on that basis. But thus far, nothing has been discovered that circumvents what we think to be the primary problem. We think the primary problem is selection pressure. And that means survival of the fittest, and that means when you use an antibiotic, the bacteria immediately begin to try to work around that antibiotic and find a resistance pathway to it. And that has led to a lowering of the use of the new antibiotics. And when that happens, there's commercial disaster for the companies developing antibiotics. And that has led, I think, to the current problem, which is that industry has largely made it very difficult, if not very difficult to find the money. And investors have found it very difficult to find the money to solve this problem. And because what they try to do is to develop a new antibiotic, and that doesn't seem to solve the problem because resistance follows the use of that new antibiotic too. So maybe for the last 40 years or so, my entire career, we've been dealing with this problem of follow-on resistance that follows use. And as we introduce ever broader spectrum and usually more potent analogs of antibiotic classes that we already know, like the aminoglycosides or fluoroquinolones or tetracyclines, glycopeptides, beta-lactams, in all these cases, resistance in each case follows use. And that levels the playing field very fast in favor of the bacteria, and we're clearly not winning this strategy. So I'd like to spend a little bit of time introducing my colleague Michael Stevens here, who's been working with us on this problem for some time and has a commercial perspective because he was the drug company guy for many years. And Michael, if you could spend a moment or two sort of talking about the dilemma of this from the drug company's perspective, that would be appreciated. You're muted.
Yeah.
Michael, you got, yeah, here you go.
Yeah, I was muted. Yes. So thank you. My name is Michael Stevens. I'm the CEO of Antibiotics. I have a 40-year history in pharma, biotech, et cetera. I've had many roles in medical, marketing, and business strategy along the way, and have dealt in the spaces of oncology and infectious disease and even clinical nutrition. So with that background, and looking at what's going on now, as Dr. Shintag said, by the time each of these new antibiotics reaches the market, we've already selected a resistance to it. And that happens during the clinical testings. And so what happens next on the pharmaceutical side is really a predictable response. First thing would be to try to develop and introduce another new analog. And that would be to offset the resistance to its precursor. But that's an increasingly more expensive way to deal with the resistance that we're seeing. And as the R&D costs rise and the market shrinks, more and more companies drop out of the antibiotic space. So there are also antibiotic stewardship programs. And these are programs in hospitals, other healthcare organizations to try to maximize the use of drugs. But what they do with antibiotics is basically they put a new antibiotic on the shelf. We're going to hold this until we need it. Okay, and that restricts its use until after the previous one fails. So you already have a level of failure that must occur before these new drugs can be used. So other things we've tried doing pharmacokinetic dosing, pharmacodynamic dosing, trying to maximize the exposure to the drug over time, and trying to hit the bug with as much of the drug as we can. And that's one attempt to try to deal with resistance pressure, but it still develops. And so what we find is that the sales of the new antibiotics that are coming out are much lower than the development costs to produce them. And so as you can imagine, big pharma is stepping out of antibiotics at a time when we really need them. I mean, the WHO projections that Dr. Shintag mentioned, those are scary numbers. And we've really got to find a way to source to get new drugs into the pipeline, get them into the clinic, and get them into the hands of clinicians. And so that's really what we're talking about here. Okay, Dr. Shintag, I'll go back to you.
Yeah, so as Dr. Stevens said, and as I think is becoming and dawning on all of us here that are in the business, and maybe some of you out there in the audience too, the current approach to antibiotic replenishment is unsustainable in the market. By definition, the financial realities of the paradigm that's just been described are preventing us from making any progress at all in the development of new antibiotics, and we clearly need something else. Now, when you think about a strategy, there's a core dictum here. As long as antibiotic use directly drives antibiotic resistance, there's probably a non-starter status for each of the ways that we have approached this problem. Number one, if you expect clinicians to use a more active antibiotic when there's an infection, they're not going to do it because they're afraid it's going to cause resistance. So they ordinarily will avoid doing that, and they'll start with the older generic drugs. Second, if you expect pharmas, or investors for that matter, to spend billions of dollars to bring out a more active analog that will then be reserved on the shelf and not used until somebody needs it, that isn't going to happen either. And the pharmas have voted with their feet on that. Third, and the current hope right now is that we expect governments to spend money on more active analogs that are going to then be placed in storage until they're needed for outbreaks. Now, there are a couple of countries that have sort of signed on to that idea. The U.S., however, has not. And the U.S. spends most of the money on this process, and a lot of the ways of looking at new antibiotics anyway. And they're just not buying it. So I think that we have to change strategy by trying to figure out how to deal with the core problem, which is use versus resistance. Now, it's my pleasure to introduce our inventor here, because Dr. Rogel has developed a direct attack strategy on resistance that hasn't been tried before. And I'd like her to talk a little bit about that, just for a few minutes on a high level here, so that we can talk about how we might use a new strategy against the paradigm of use versus resistance, and see if we can't develop what I might call an antibiotic sustainability option. Snezna?
Thank you, Dr. Shentek. So yes, my name is Snezna Rogel. I am just very recently retired from being a professor for 26 years at this small New Mexico STEM university, where, in addition to teaching cell and molecular biology and biochemistry and some microbiology, I taught students how to conduct research, right? Offered them hands-on experience, which not only allows them to learn how things really work in reality in science, but permit for their exploring their passions and their creativity. So over the past 26 years, I've had them work in my and my colleagues' laboratories, pursuing the kind of things that I found I really care about, and that is cancer. I've done cancer research since graduate school and then at MIT and Harvard. And then parasites that plague primarily the developing world, like intracellular protozoans, and then, of course, bacteria that plague the whole world and are threatening to become worse, particularly drug-resistant organisms. And we've realized early on that life, small and large, is a combinatorial biological process, right? Life emerges from these combinatorial processes. It's an epiphenomena. So the solution to AMR, antimicrobial resistance, is not likely to be a single stroke answer. It's likely to have to be combinatorial as well. So for many years now, along with my undergraduate and graduate students, we've tried to identify mechanisms whereby we could selectively as possible eliminate bacteria. And indeed, bacteria, like in a slightly different way, mammalian and eukaryotic organisms, possess a mechanism with which they detect DNA damage, right? And they first try to repair that damage. But if that does not happen, bacteria kick on a mutator response, one that's sort of a Hail Mary response to, we're being damaged. And of course, many antibiotics, either directly or indirectly, are DNA damaging. So they respond to stress by creating more and more mutations. And most of those, by the vast majority of those mutations, are useless. The organisms die. But those that do provide resistance, those provides resistance to the stress, then proliferate because their mechanism is now written within their genome. So targeting the mechanisms whereby the recognition of this DNA damage, of the stress, of the need to turn on the mutator response is muted in bacteria alone, appears to be an ideal approach for we could thereby prevent the bacteria from responding by generating more and more mutations. And I can elaborate on that more later, but I think we need to go back to the sort of the overall ideas that Dr. Shentek is expanding here now.
Thank you very much for that discussion and for the reality check here that we've all had to go through, which is that it took an inventor who has been working on this thought process from a different perspective than most people. Rather than attacking the question of developing a new molecule of the same class, what has been done here is we figured out a way to deal with the way bacteria develop resistance and stop it. And that means that we now can go back and think about this from the perspective of maybe we don't need to develop new antibiotics. We just need to figure out a way to stop the old ones from being resistant and avoid having to go to a new one. So the marvel of this strategy is that sustainability now can be defined as let's figure out a way to recover the antibiotics that we've already lost because selection pressure has been, not their friend, by using this new class of molecules as a helper. And so just to recap here, this helper strategy, if it costs billions to develop a new antibiotic analog, which is not fixing the selection pressure, and it costs a large sales price to recover those development costs, and thus a high price for a new drug that doesn't solve the problem. And that reaction to that high price makes it unsustainable for the company to develop new antibiotics. We have to think about using this strategy to restore the antibiotics we've got and see if we can get some more timing out of them. And Big Pharma with its global logistical reach has to think about it from a different perspective. We need them to come back, as Dr. Stevens said. And we have to deal with the fact that if we're going to get them back, we have to get them convinced that, they're probably right by voting with their feet when the idea is that just developing a new analog of a drug we've already had for a long time is their only choice. But they're going to have to make some compromises here. And that is they're going to have to work with drugs that are not drugs that they still have under patent. And that's not a pharma strategy that's very popular. And perhaps Michael can spend a moment or two talking about how they usually react when you say something like this. And then we'll talk about how we think we might be able to talk them back into doing it after that. So I'll be back, Michael.
Yeah, there's really two problems from a pharma standpoint. The first is, and this is true for all drug classes, there's really limited financial return when you start investing in generic drugs. Okay, there's no real bang for your buck, if you will. And so that precludes them from putting a lot of money on the table. But the other problem that we have with antibiotics is that when a doctor writes a prescription, it's a very short-term treatment for that patient. It'd be like a Z-Pack, or something that's going to be, 10 to 14 days, and then you're done. Well, what pharma wants to see is they want the next Lipitor. They want something that when that prescription is written, that patient's going to be on it for years. And that's true with insulin, like I said, the statins, and other long-term drugs. So there's, that's the problem. Now, the one exception to that from an antibiotic standpoint is what happened with the HIV drugs. Okay, in that case, unfortunately, because we don't have a cure, patients do stay on those drugs a long time. And so pharma did step up to the plate. And over the past 20 years, there have been a number of new drugs, and most importantly, new combinations of drugs in the same capsule, the same pill, that have gone through this whole process and are now on the market and have made HIV a really manageable disease by and large. And so what we have to do is come up with ways of showing through the demonstration that we've just said, how we can take these old existing drugs, combine them with the new drugs that we have at Stymbiotics, and then create those drugs that are going to be more valuable for a longer period of time, even though they're multi-source.
So that actually is a strategy which pharma has succeeded with in the past. There's a time when beta-lactam antibiotics were dying off as well, and it was discovered that you could combine an inhibitor of beta-lactam, beta-lactamase, the enzyme that bacteria create to destroy it. And when we did that, we made combination products that were very successful in the market and are successful to this day. They're much more active beta-lactams, and in fact, the newest antibiotic combinations that are being sold by pharma now, the ones that have been developed as improved analogs, are all beta-lactam, beta-lactamase inhibitor combinations with the newer beta-lactams. So that works, but the problem is as has been said already, the problem is that that fixes only the beta-lactams, and that doesn't fix the selection pressure completely, but it helps. We need something like that for drugs like vancomycin, for aminoglycosides like tobramycin, for the polymyxins, for the macrolides, for the tetracyclines, and all the other drugs where nobody's ever managed to find an inhibitor like the beta-lactamase. And what we're trying to do here, as you can see, is we believe that we've got that. We believe we have a class of inhibitors now which can be combined with these old drugs and restore them just like happened with the beta-lactamase. And Symbiotics, therefore, has a new approach to sustainability, because what we want to do is to find some partners who then can help us to restore these old antibiotics, even around the reality that they're generic drugs, but this new molecule plus the generic drug may very well be a patentable combination. And that's the basic core strategy that we'd like to see happen here, because we can't continue doing what we're doing. And this is the only thing so far in 40 years of work in this area that I've seen that looks like it has some promise in fixing this problem on a core basis. So that's our story. And thank you very much for your attention. And I think we have a little time left for Q&A. So I'll turn back over to our moderator.
Thank you very much. This is absolutely fascinating, and it's a really fantastic job you've done. You've managed to cover quite a complex subject in very succinctly and to the point, involving the clinical practice and the investor practice at the same time. I have a few questions, if you don't mind, that just came to my mind through this discussion. And I'll start with you, Prof. Sentek. So you're thinking that this new approach might buy us some time then by extending the life of the antibiotics we'll already have, and then that makes it also attractive for the industry to come and invest.
Yeah, certainly we aren't thinking that we've solved the problem forever. Bacteria are very clever, but we have done quite a lot of work on this idea that how long do you get before you get resistance to this new combination, and it doesn't happen easily, if at all. And we've done some of those studies already where you do something called a serial pass experiment, and that's been quite successful so far, as we don't seem to have resistance to the resistance treatment. But you never say never in this business.
Absolutely true. A question for you, Dr. Stevens, because the industry, you've mentioned the industry a few times, and it does not usually invest in improvements to antibiotics that are already generic. And now with your discussion, you imply that the combinations with the Steambiotics platform compounds, they would breathe new life into these old drugs by preventing resistance. So how should we talk with pharma? Because this is a new concept. It's a paradigm shift to how they've been approaching this field for many decades.
Yeah, it is a paradigm shift. Basically, by coming out with a combination of our drug with, as Jerry said, with tobramycin or with vancomycin or whatever, you can get an intellectual property around that. And so, you know, the base drug is already on the market. That work has been done. You do have to do the work to show that the combination is effective. But what we're seeing is that as he said, it's really interesting. We can take a drug that is resistant or a bug that is resistant to a specific drug, and we can basically revert it back to wild type. And so that basically turns that drug back on. And so this is a cost-effective way for society to be treating these infections. Basically giving new life to old drugs. And so we have to convince pharma that that's in their best interest too, to be providing such products. And we still can't get over some of the other problems, like the fact that it's relatively short term, but it's also not a very high bar like you have for developing a totally new molecule, which is usually just a copy of a molecule that already existed. Here we're taking existing molecules and basically turning them back on.
No, this is absolutely fascinating. Uh, Perhaps I should have asked ladies first, but I've just taken it in the way that we spoke.
Right. All right.
So just coming to you, Dr. Rosal, your invention of these helper molecules implies that most of the generic antibiotics we have could be paired with a helper molecule from Stimbiotics. So can we study these aspects quickly? Because the current pipeline is quite a long one anyway. So can we do that quickly to define the best match before going to clinical trials? Or have you been doing that in your lab? How much of that work has already been in place?
Yeah, thank you for this question. It kind of rests on what is quickly, right? So yes, we've been working on this for over a decade. This family of compounds has been in the works at New Mexico Tech slowly because of our constraints over this past decade, but we did manage to develop a series of leads that work by themselves directly against gram positives and gram negatives. And then we discovered that, in fact, they work very well in combinations. And as was just pointed out by Michael and Jerry, what we can do is take a drug-resistant gram-positive organism, combine it with a Toby or a Vanco. It depends on the organism and it depends on our lead, but we can, in combination, in a drug-resistant organism, bring back the resistance to while neither of the compounds works by themselves, at concentrations that neither of the compounds works by themselves, in combinations, we can kill that organism.
Okay. So I'm thinking, as in clinical practice, that would come down to a matrix system.
That's right. And so we have created these matrices. We have 5 different classes of antibiotics. We have a dozen of our leads, and they work together in certain combinations. Not all the combinations, and not all combinations are required, because some, MRSA may or may not be resistant to oxacillin or another antibiotic, but together they do work very well and bring down the resistance to what is clinically acceptable, both for the standard antibiotics and what is, best we can tell, safe for our own potentiator. And we have compounds that work against gram negatives as well, both by themselves and alone in combinations with things like colistin and Cipro. So yes, it is a complex matrix, and perhaps that's why it's taken us so long. But had we or if we can get funding in order to address this as a combinatorial process, inescapable combinatorial process, things can move rather quickly, because now that we know that this is doable, now that the window is open, we can approach it systematically and get the results, meaning both in vitro and then in animals and potentially clinically, way faster than we've gotten to this point right now.
Oh, that's very clear. Thank you. Just a final question, because I see we are close to the time. Dr. Sentak, we haven't mentioned if any investors are interested in participating if any of the audience is interested to contact you and take part in your journey, how do they contact you for further discussions? if you could just give us a point of contact and a way.
You're muted. Jerry, you're muted.
Yes, sorry, we're all on LinkedIn. We're all out actively trying to talk to investors, and you can find us by email, just with the names, I think, in most cases.
Yes.
We're not trying to hide here. And I did ask for screen sharing, but I didn't get the permission to do that, so I can't put up our contact information directly for people. But I do believe that if you hunt for any one of us on the web, you very quickly find it. Thank you so much for the opportunity to address this question to our moderator and to the group that's putting this on, that we really, the live streaming service that we've done. It's been a pleasure.
I can
Yes.
link that.
Thank you very much.
Thank you. Thank you all, and thank you to the audience. Thank you and good night.
Goodbye.
Good
Bye.
night.